Hi everyone, thanks for joining this afternoon's webinar. We're all good to start here, but I just want to give it another minute or two to make sure all participants are in, so just bear with me for a minute. Okay, I think we're good to go now. Okay, so hi everyone, my name is Karen McCarthy and I'm the Technical Sales Manager here at Adhesives Research. I'd like to welcome you all to today's webinar on how pressure-sensitive adhesives can improve the rapid scale-up of microfluidic devices. Sorry, apologies. Okay, so for the structure of today's webinar will be brief introduction given by myself, just in terms of why we're bringing this content to you now. It's just lining why there's a growing demand for microfluidic technology and the kind of things our customers are looking for in PSA tapes. This will then lead into a presentation by Tom Gordon on the rapid prototyping of microfluidic devices with pressure-sensitive adhesive tapes, followed by Q&A. But before that, I just want to quickly run through some webinar housekeeping with you. Okay, so you'll all be in listen and view only mode, and you will not be able to activate your webcams or your microphones. The Q&A session will be held at the end of the presentation, so please submit your questions through the Q&A feature. You're allowed to send anonymous questions and allowed to view all questions, but you cannot upvote. And any unanswered questions will be followed up after the webinar. The recording can be accessed on demand post the event, and please use the raise hand feature if you need any special assistance. Okay, so why now? Because there's clearly a growing demand for microfluidic technology and devices in point of care diagnostics. Market research has estimated an annual growth rate of about 11.25% from the year 2020 up to 2027. So to put a figure on this is a growth of about from 12.3 billion euros rising to an excess of 25.5 billion euros by year 2027, so quite considerable. So what is driving this market? Well, first and foremost is the increasing incidence of disease and the need for rapid and accurate tests for diagnosis. This has clearly been the case within the last year with the onset of the COVID pandemic, and there's been an earnest need for the speed up in the development and manufacture of COVID test devices. There's also an increasing demand for low volume sample analysis. So low volume sample analysis is attractive in terms of it reduces the amount of reagent used, it reduces the amount of reagent waste, but it also helps to preserve samples that are difficult to produce. There's also a desire for multiple analyte detection in one device. So things like lab on a chip or microarrays where you're testing for numerous biomarkers in one single device. So this enables a much faster analysis and response time when you're comparing it to the conventional laboratory techniques. So what are the restraints? Well, first off, I suppose depending on the specific application, the workflows can be very laborious. So to design an integrated test chip that encompasses the entire workflow from start to finish can be extremely challenging. There's also high costs that are typically required. So for the capital equipment needed to fabricate such devices, so things like equipment needed for lithography, for machine lasering, for hot embossing, these can be quite expensive. And if you don't have ready access to this type of equipment, it can rule it out for some customers. This is where we see the desire for adhesive tapes. So these can be used quite simply and easily and will overcome that capital cost need. So again, within the last year, we have seen a dramatic change in terms of our customers and how they're shifting their focus and priorities during the pandemic. whether this be through adapting existing platforms or developing new platforms. They're looking for solutions to meet these objectives quickly due to the critical demand for these COVID test devices. So when our customers are developing devices with the use of adhesive tapes, they're looking for solutions that will be, I suppose, biocompatible. So you don't want the adhesive interfering with either your test reagents or the biological sample. So you need a medical grade adhesive that's proven biocompatible. Also looking to bond dissimilar surfaces. So depending on the device, you could have a low surface energy substrate and a high surface energy substrate, and it can be difficult to bond these two surfaces together with one adhesive. So the solution here would be to use a double-sided adhesive with a low surface energy adhesive on one side, such as a silicone adhesive to bond to the low surface energy plastic, and a high surface energy adhesive on the opposing side to bond to the high surface energy substrate of your device. So this allows the dissimilar surfaces to be combined together. Also looking for low residuals. So if you've high residuals within the adhesive or the construction, they can outgas over time and interfere with the reagent chemistry. So it's critical to keep these low. Low autofluorescence can be desirable depending on your method of detection within your device. So obviously, if your method of detection is fluorescence, you don't want any background fluorescence coming from the adhesive construction. Looking for tight thickness control. So if the adhesive tape is to be used as a spacer within your device, the thickness of this tape would dictate the height of the capillary and thus the sample flow or the sample volume in your device. So it's critical that this is maintained at a tight thickness control and is consistent batch to batch. Precise contact angle is desirable if the adhesive tape is going to be used to modulate the flow through the chip or the device. So for example, if you're looking to speed up the flow of the sample through your device, you're going to need a hydrophilic adhesive with a low contact angle that will wick the sample through quickly. Equally, you might be looking to slow it down somewhat, and then you'd have an intermediate contact angle, possibly a hydrophobic adhesive, in instances where you've possibly reaction chambers within your device and you need the flow to be slow in that area. And then finally, the use of adhesive tapes for device fabrication enables rapid, hassle-free production. There'll be more on these topics in Tom's presentation, which is up next. So with that, I'll hand you over to Tom.
Hello, my name is Tom Gordon. I'm the Healthcare Innovation Group Leader at Adhesives Research. Today I'm going to be discussing the rapid prototyping of microfluidic devices with pressure-sensitive adhesive tapes. So Adhesives Research has a 25+ year history of providing tapes to the medical device industry, including in the area of microfluidics. We began with developing tapes for lateral flow immunoassays, so things for pregnancy testing, for infectious disease, as well as for the identification of chemical contaminants. We then moved into the area of microfluidics and devices such as blood glucose test strips, blood coagulation devices, the detection of prostate-specific antigen, as well as forensics and drugs abuse. And we continue to innovate for the future in areas such as continuous glucose monitoring, gene sequencing, PCR testing, and organ-on-a-chip or cell growth types of platforms. And before I get into the rest of the presentation, I'd like to briefly review the types of pressure-sensitive adhesive tapes that are available in the marketplace. So one would be transfer films, in which a single layer of adhesive is placed on a release liner. This can be used to bond two materials together, and this is your best option for forming a very thin channel height with a pressure-sensitive adhesive tape. There are also single-phase tapes in which a polyester film or another type of backing can be used to support the tape. These would be bonded or only on one side, and they would have a release liner protecting the adhesive. And then there are double-phase tapes in which the carrier has adhesive on either side, which is protected with a release liner. Microfluidic devices are commonly prepared through process known as soft lithography. It begins with the traditional photolithography approach, in which you take a silicon wafer, you spin coat SU8 or another type of epoxy material onto it, and then through a lithographic mask and UV light, you form your master device. So this type of master device can then be replicated numerous times in the soft lithography process. And that involves essentially casting PDMS onto the surface of the master mold, curing it, and then removing it from the surface. And then applying it to a glass slide surface after plasma treatment. And this is a highly precise fabrication method. However, it requires this microfabrication facility, which is expensive to access or expensive to build, and requires that plasma treatment in order to get good bonding. And altogether, it's costly and time-consuming process. So researchers have developed tape-based approaches to do rapid prototyping of these types of microfluidic devices. And that can begin with the design of an AutoCAD design of your microfluidic channel. That can then be easily cut with a plotter cutter, which is often referred to as xerography, using a razor to cut the shapes of the microfluidic channel, or through laser cutting of a tape into the appropriate microfluidic channels. That tape, that after it's cut to the appropriate microfluidic channels, can then be assembled with a base and a cover to form your microfluidic device, simply with the application of pressure to bond the adhesive. And then finally, once this full device is formed, it can be disinfected or primed in whatever way is necessary prior to the introduction of the biological materials. A recent report from Rothbauer and et al. This in Scientific Reports discussed the characterization of 4 biocompatible pressure-sensitive adhesives for this tape-based approach for lab-on-a-chip and organ-on-a-chip systems. And if you look at the approach that they're taking, it uses much lower cost equipment relative to the microfabrication path that I discussed on the previous slide. It can cost $1,000 to $20,000 for one of these home hobby types of plotters, and up to $100,000 for a more professional, more capable instrument. And you can also get into laser cutting with between $20,000 and $150,000 worth of investment. So it's not too hard to get the equipment and not too expensive to get the equipment required to cut these microfluidic channels. And I think even a better advantage is the total time spent between designing the AutoCAD file to producing your microfluidic device and disinfecting it is a total of 1 to 3 hours, which is substantially lower than what is required to access the microfabrication facilities and produce the full device through soft lithography. Other interesting findings are that acrylic-based PSAs were found to have lower vapor and moisture permeability than PDMS, which for certain applications such as organ cell growth is very important. You don't want to have very high moisture permeability. Acrylic adhesives were also found to support cell growth on their own, which is a problem for other types of materials used in microfluidic devices. And then the membranes and scaffolds can be introduced into the device on their own through the use of the PSA tapes, which allows for the support of organ cells and other types of cell growth. I think another major advantage of this type of approach is it allows for the bonding of dissimilar materials. So PET, PDMS, PMMA, COP or COC, all these materials, polycarbonate, polystyrene, can all be bonded using pressure-sensitive adhesive tapes, as well as the membranes and scaffolds, as I mentioned. And that can provide a huge advantage. For instance, I show an example here where polycarbonate can be bonded to polydimethylsiloxane or PDMS through the use of a differential adhesive tape, where an acrylic adhesive is designed for the bonding to polycarbonate, a silicone adhesive is used for PDMS. This is something you can't accomplish using that traditional approach. However, there's some very critical aspects of a PSA tape that need to be taken into consideration when you're selecting a tape. The first would be the biocompatibility of the tape. adhesives can be tested for their accordance with ISO 10993-5 and -10, cytotoxicity, skin irritation, skin sensitization, and that gives you an idea of whether the tape will support or interfere with biological activity. Related to that is the residual content in the adhesive. So after the adhesive is manufactured, it should have low residual content of volatile materials. And I show an example here of R-Care products, acrylic tapes, relative to a competitive tape, showing the low outgassing that we can achieve in our tapes by designing the process to produce them in a way that's going to drive down residuals. And in addition, the uniformity of the tape is very important, particularly for microfluidic devices. Your channel height is essentially defined by the thickness of your tape. So specialized coating equipment is needed to get very uniform thickness of adhesives for this type of application, and repeatable adhesive thickness as well. Another important aspect to consider is moisture resistance. Adhesives should provide a moisture or fluid resistant barrier. Particularly, this is important in multi-channel devices where you do not want fluids to be interchanging between the multiple channels of the device. That also includes, you don't want air or other contaminants from getting inside as well. Autofluorescence is also very important to keep in mind if you're doing an optical type of test. The tape cannot fluoresce excessively in the region of interest. So we show here numerous adhesives produced by Adhesives Research showing that they have relatively low autofluorescence at some common wavelengths of excitation, 485 nanometers and 532 nanometers. And then finally, the cuttability of the tape is very important. So if you're going through either a plotter cutter or other types of cutting, laser cutting approaches to define your channel, the modulus of the tape is very important to consider, that it's going to cut well, it's not going to smear on the edges, and it's going to make a very well-defined edge for your microfluidic device. And a real advantage of this rapid prototyping using tape approach is that it's also easily scaled. So you can take that same AutoCAD design that was developed for use in laser cutting or the plotter cutter and introduce it right into medical grade converting, in which you would cut the tape roll to roll and wind up a converted roll that has your microfluidic channel already cut into it. That can then be used in a reel-to-reel type of manufacturing environment or reel-to-chip manufacturing environment, in which all the required components for the microfluidic device can be added in a reel-to-reel environment. And that is the best approach to do large-scale production of microfluidic devices. And the various options to cut these channels would be things like laser cutting, which I mentioned earlier, as well as rotary die cutting or flatbed die cutting, which can cut the same shapes as produced with these other approaches. Adhesive Research provides some really unique technologies in the area of microfluidics. One will be our hydrophilic pressure sensitive adhesives. So these are really unique products to provide both pressure sensitive adhesive strength as well as hydrophilicity, which allows for fluid flow and wicking of fluids into the channel. So we provide both a 93049, which is a single phase tape, and a transfer film 93450 for these types of applications. We also provide high uniformity double phase spacer tapes to define the channel of a microfluidic device. So these include various different acrylic adhesives, heat sealable adhesives, as well as different thicknesses of carrier that can be used to define your channel height as needed for a particular device. In addition, we offer numerous transfer films at various thicknesses. So these can be silicones, these can be acrylic-based tapes that provide very strong bonding to dissimilar materials, as I mentioned earlier. We also offer products that are optically clear, so they can provide a very clear path for either identifying fluorescent compounds inside the material or looking at fluorophores or for exciting those materials as well. In conclusion, adhesive tapes provide a low-cost platform for the rapid prototyping of microfluidic devices for a multitude of applications. However, medical grade tapes must be properly selected, and there's some important characteristics to consider, such as the biocompatibility, the residual content, the thickness and uniformity, the moisture resistance, the autofluorescence of the tape, as well as the cutability of the material. And these tape-based microfluidics can be easily scaled to high volume, reel-to-reel processing. Adhesives research provides a lot of value to the customers who partner with us. We provide design flexibility, we provide very fast development capabilities, and it leads to extraordinary outcomes for our customers. And so, our R&D department partners directly with our customers, are the engineers who are designing microfluidic devices, so we can provide the ultimate solution that will solve your problem. And at the bottom of this slide is shown some of the manufacturing and production sites that we have, both in Pennsylvania, in Ireland, and we have sales offices in China. And we also recently brought online medical converting in Glen Rock, Pennsylvania as well. Thank you for your time, for being involved in this presentation. I invite you to contact us at Adhesives Research. We'd love to partner with you to solve your problems in microfluidics, and we have a lot of unique products to offer in this area. So thank you.
Great. Thank you very much for your attention this afternoon. So we can now start the question and answer session. Actually, during the presentation, 2 questions came in, so I guess we'll start with those. So we can start with Anuka's question. Sorry if I didn't pronounce your name correctly, but the question is, do you think it's okay to use devices right after assembling, or should it be settled for some time before first use? This is a good question. For most pressure sensitive adhesive tapes, it's best to leave a little bit of time for the adhesive to reach full bond strength. So the amount of time that takes varies somewhat from adhesive to adhesive, but typically within 24 hours, you can expect to see the maximum bond strength. Some tapes may require up to 72 hours, but typically, 24 hours is best. I would say it doesn't mean it couldn't be used right away. It's sort of when you could, there's no other problem other than you may not have the ultimate strength in bonding the 2 materials after, immediately after they're bonded. And then maybe, Karen, maybe you want to take the second question of where can we buy your products?
Okay, sure. So you can buy the product essentially direct from Adhesives Research. So if you let us know where you are, we'll put you in contact with the salesperson for your region. You can discuss your requirements with them, and we can deal with it from there.
Thank you. Looks like there's 2 other questions just came in, so I need to read. Shane O'Sullivan has a question here. It's a little bit detailed, so let me have a chance to read through it. They're using RCare 7840. Had some issues ensuring correct seal, PSA to a silicon chip. Yeah, so they're having some issues with the fragility of the silicon that they're dealing with. When applying the adhesive, it's snapping or cracking. Yeah, so I guess one sort of related to the first question, you can apply minimal force, I guess, to bond an adhesive and then give it some time. It has a tendency to flow with time and get better bond strength. You may also put the adhesive in like an autoclave, something that not applying any humidity, but simply just pressure, raise the pressure in there, any air bubbles or air separating the the adhesive from the silicon chip should be pushed out. So that's, I guess, 2 suggestions I would make. So hopefully that answers your question. So Amulya Raj asked, is it suitable for styrenic-based copolymers? So yeah, so I assume what this means is, do we have adhesives that are suitable for bonding to styrenic-based copolymers? And I would say the answer is yes. We actually make styrenic-based copolymer adhesives. They're not commonly used in microfluidics. We tend to use acrylics and silicones most, but we make rubber-based adhesives. I would say silicones also would have a good chance of bonding well to styrenic copolymers. So yeah, something we can certainly try to give you samples for. And then I guess we have a question here about maximum pressure that these devices can handle. I would say that really depends on the design of a microfluidic device. So we can give you ideas of how much bond strength an adhesive can provide in terms of shear strength or lateral shear strength. So it really, but I think the size of your channel and the really is going to also impact how much pressure can be applied within the channel. But generally, I mean, there's many microfluidic devices made with pressure sensitive adhesives, but I don't know if any are very high pressure, but I know pressure can be applied certainly. Let's see. Yeah, so a question from Mike, who I've never met, but familiar with. You have a question about G prime versus G double prime. Does the temp sweep predict die cutting performance? Yeah, I think at mostly we're talking about at at given temperature, you want higher modulus in general for die cutting. So that was just sort of showing an example of how you can measure the modulus properties of an adhesive. Generally, if we're comparing two adhesives, we want higher modulus for better die cutting performance. Ian is asking if a microfluidic channel or channel required functionalization, such as albumin coating, what type of adhesive would work and not work. It's an interesting question. So I assume from the question that we're asking if we're bonding to the surface of a albumin coated surface, what type of adhesive will work for that. I can't honestly say we've tried that particularly. I would, certainly we can give you a range of adhesives to try, whether it's acrylics. There's different aspects of the adhesion. Some of it is physical, so adhesives will bond to anything. Some of it is chemical. So is there an interaction between the albumin that could help get better bond strength? So that's something I think we could talk offline if you want to contact us. About tape cutting, what is the minimal size of the channel? So that's a great question. So with die cutting, you are unlike you won't get as fine of features as you can get with, say, lithography. So you're usually talking about, I'm not sure if I can give you an exact precise the minimum, but we're usually talking more about a millimeter or so in width, rather than down to hundreds of microns. That's not easily done with die cutting. Laser cutting, you can get a little bit better than die cutting. But yeah, we're generally not talking about as fine of features as you can get with lithography. So thank you. That's a great question. I think the xerography versus laser cutting question there, that's a great question. Joan asked a sort of similar question. I think, like I said, I think normally with xerography, and of course it depends on the equipment there, but I would say more like a millimeter size gap is probably what you're looking at there. Laser cutting, I think you can get slightly less than that. Yeah, so Shane asked about the follow-up question about the ballpark time. So I would suggest, again, sort of like the earlier question, I would say 24 to 72 hours is what I would suggest in terms of getting the best bond strength you can. Great. So Allison asks, would you give ranges for heights of the final adhesive layer when you add the glass surface to form a microfluidic channel? Right. So in terms of the heights, so like I said in the presentation, so there's transfer films and then there's double face tapes. The thinnest, well, we can make a transfer films relatively thick, but normally we're talking about 12 microns. You know, usually 10 to 12 microns is the minimum height. We could potentially go lower, but that's around where we are today. And then up to really whatever height you want, depending on the carrier thickness, or we even have some transfer films that are at 250 microns. But yeah, we can go relatively thin, but then thickness is generally easier, where we add a PET layer or something like that. It's 100 microns or 50 microns. We can build up the thickness. So here's a question about the consideration, and I think this question came in earlier about the criteria for adhesives during thermocycling, specifically PCR. And that's a great question. So you need to have a PSA that's resistant to the cycling. The one thing that's really nice about silicone adhesives is that they tend to have less of a change in modulus over time, or by changing temperature. So as there's fluctuations in temperature, you don't see as much of a modulus change. So that's a great aspect of silicones, why they're often used in PCR, but acrylics can also be resistant up to, the temperatures around 100 C. So you need to find an adhesive that has sufficient shear strength at elevated temperature, that it's not going to debond or peel strength and shear strength at elevated temperature. And then the question came to how thin can you make a double-sided film? So I would say normally our minimum would be something around 37.5 mils. So if you have about a half, so you have about 12 mils, 12.5 mils, 12.5 microns of adhesive, 12.5 microns of a carrier, and then 12.5 microns of another adhesive. That would be relatively thin for us. We've been pushing that limit to lower and lower, but you're usually looking around, I would say 30 to 40 microns for a double face as the minimum. And then one other point about the PCR, you also have to consider all those things I talked about in the presentation. So residuals, autofluorescence is often really important for PCR. So not all tapes are made the same with respect to that. And also, I think another consideration for PCR is, DNase, RNase, those other components that can interfere specifically with the PCR test. So, we have clean room based coders. So that's something you want to have in the tape, is that it's being made in essentially a clean room environment, so it's not being contaminated. Great. How does time affect hydrophilicity or using for several times? Yeah, that's a great question. So for hydrophilic adhesives, generally they're single use. I think you could get multiple uses out of it, but it's not generally done with multiple uses. And then in terms of how does time affect it, I mean, we have done lots of shelf life studies on our adhesives, and they're stable certainly for at least a year. We have a lot of customers who have used these tapes out for multiple years in terms of shelf life for their product. So they're stable once assembled into a microfluidic device, but generally it's more of a one-time use type of thing. Okay. To make thicker, deeper channels, can we stack these adhesives? Yes, you can. So If you need to build up a layer, that's definitely a good way to do it. Again, you want to allow some time for the layers to bond to each other. And I would say the only thing I can think of as a possible issue there is if you're trying to stack an acrylic adhesive with a silicone adhesive or something that's dissimilar. That's where you really want something in between, like a polyester or something, because the adhesives may not bond well to each other in certain cases. Okay, so are there already any IBD related products which apply your tapes in contact with full blood? If yes, can you name the applications? If no, so yes, we have lots of products that are in contact with full blood. So one good example I would say is blood glucose test strips. Been involved in that business for many years. So yes, in that case, you're in contact with full blood. Maximum contact with is there maximum contact with liquids or samples? Will there be a leachable event taking place after some time? Yeah, so I mean the leachables in our tapes in most applications or for most of our products are very low. So you're talking about ppm levels. So and generally if you're making like a channel, you're really only contacting the edge of that adhesive for some amount of time. So in some cases there can be leachables we can essentially design the adhesive around whatever your test is, that it's not going to interfere with your test. But yeah, I think there's no maximum time there's no specific time of exposure that will cause the adhesives to fail. Generally, they're stable in the presence of normal fluids. It's when you get to higher temperatures, some type microfluidic tests require base or solvents or that type of thing to go through the channels, and we have adhesives that are resistant to that as well. But that's more of a challenge, I would say, than just water-based fluids. Okay, it's like a lightning round here. Alejandro asks, in R&D, we are simply bonding your tapes manually with our hands. Do you recommend some specific pressure to obtain a better performance of bonding? But yeah, so that's a good question. The best thing I would say if you're starting to get into bonding adhesive tapes is to get a roller. It's great to apply firm, even pressure as you're assembling devices. And then, I would say, if you're applying something like 10 PSI, 10 to 30 PSI, I mean, you can also invest in like a laminator. So basically just be two rolls that are kind of two rubber rolls that are in contact with each other, controllable pressure, controllable speed is something that you can also, start to play around with the pressure and speed settings to get the ultimate bond strength. Or better, I think what you really want to eliminate is any air bubbles or anything like that. And that's where a roller, where you're placing the one side of the adhesive down and you just roll the rest on, is the best way to get a bubble-free lamination. Nasser asked if we use a tape as a base for microfluidic strip, would the glue used in the tape interfere with the fluid or any of the particles flowing in the channels? Yeah, so that's a good question. So you can sometimes get non-specific binding of certain materials in the fluid, but generally, one thing that our hydrophilic adhesives are used for is to really help wick fluid through a channel. So you can look at hydrophilic adhesives for that purpose. But generally, like I said earlier, I mean, certainly some tape can interfere with, with your tape, with your test, if it's leaching out things, if things are coming out of the surface, that could be a problem. But generally, we don't see that issue commonly. So question again about where we can find your products. Do you have any distributors? So generally we do, well, maybe, I don't know, Karen, if you want to take that question.
Sure, you're asking if we have distributors in England or Europe and whether you can purchase immediately. Well, the distributor in Europe, I guess, would be the site that I'm based in. So we have a plant in Ireland, numerous salespeople here. So I think the best thing is to reach out to either me directly or one of the other sales associates here. I think if we should have your details after this, and I'll reach out to you directly and see what products you need, what you're interested in. If it's readily available, we should be able to supply it soon. So it depends on what exact product you're looking for.
All right, great. And I think we're approaching the end of the time here. I think there's a question here about recommend cutting products. I assume that means like maybe the xerography type of thing. I don't have a specific recommendation, but we can probably provide some suggestions. And then I would really recommend if you look into xerography and microfluidics. There's some really good papers out there describing these processes. I covered one of them here in the presentation. That one provides specific equipment that you can try to purchase to do this. All right, and so I think we're out of time here, unfortunately. I really appreciate everyone's engagement today on this. This has been really great to see all the interest and all the questions come in. So any remaining questions, which I see there's quite a few here, we will follow up on and get you an answer. And thank you so much for joining. And if you have any additional questions or inquiries, you can also email marketing@arglobal.com, and we'll be following up with everyone who is in attendance through email in the coming days. So thank you very much for your time and attention.
Thanks, everyone. Bye.